So that's actually a really good question. It does take a lot of attention and work. So here at my institution, we don't have dedicated readers for prostate MR. Anybody who reads abdominal MR will read prostate MR as part of that. So maybe we have about a dozen readers. In terms of just evaluating the quality of this, there's numerous components of it. We do have a kind of a weekly MR conference where we'll review cases from the week, challenging cases with interesting follow-up. That's actually a great opportunity to bring forward to the group for discussion some of the more challenging prostate MR from that week. We're going to have some banter, some discourse, and it can be a very good learning opportunity. In addition, anybody who reads a prostate MR and that MR goes on to a targeted biopsy, we have a digital mechanism put in place actually by a colleague in my division where the path results from the targeted biopsy automatically gets emailed back to the radiologist who read the case. If I read a prostate MR, say this week, next month I get a biopsy, I automatically get informed of the biopsy results. I don't need to look it up. I don't need to be tracking it. So if there's a discrepancy, or whether it's concordant or discrepant, I'd go back, see the MR, see the path. That can be a good opportunity to learn. And then in addition, I kind of help oversee the overall prostate MR program at my institution. So say we have a new reader, somebody new joining the faculty. When they first come, we can kind of go over our approach, how we read them, kind of our protocol, our template. At least in the early stages, do a little bit more closely, intermittently reviewing some cases with the person, some feedback, just kind of as a bridge. When we have a new person join, I can be a little bit more of a resource and kind of see how people are doing and give some feedback that way. And I think overall we're doing well. So it's definitely a tough issue. That's some of the ways we've addressed it. I know other places have their own solutions as well.
So at Hopkins we have 7 radiologists who read body MRIs, and when they cover the MRI service, they do read prostates as well. And 2 are in the community radiology group. And there will be 3 new coming soon with the new academic year. So we'll have 10 prostate MRI readers. The way we're trying to transition faculty to feel comfortable with reading MRI is all of us in body imaging have been involved since the beginning of MRI, but the new faculty I encourage to participate in our monthly conference where we have the MRI pathology correlation, where as a group we review cases that have MRI, of course, with the biopsy results, and we try to score de novo the cases and discuss how we apply PIRADS to each case, and they look at the results and then look at the fusion captures that were taken to see if there's discrepancy, if there is a possibility that there was a misregistration, versus if there was a discrepancy related to the PIRADS scoring. So we have this monthly conference. It's a WebEx type of conference, so those who are remotely or not on MRI service participate, and that's our way of transitioning. And also I'm available to all faculty who have questions, so I take emails and calls and look at any case that they may have questions about. So I think this is sort of a collaborative approach, and we feel very good. We studied our results as a team. We're doing relatively well in terms of consistency.
At Cornell, I'm very fortunate to have almost a dozen of my fellow section mates who read prostate MR with me. A number of them have been reading it for some time. At UCLA, though, we restricted it to only 3 of us that read prostate MR. That was important early on to ensure that we had a uniform level of quality. But with the use of PIRADS, what we found is that the interpretations are fairly standardized. Bringing my new faculty up to speed involves figuring out how they've been trained to ensure that they've been taught the current PIRADS assessment system, and then to go through cases, usually those cases that go for image fusion targeted biopsy that I would contour myself. That gives me an opportunity to review their reports and their interpretations.